Have you ever looked in the mirror and wondered if time could be slowed down, or perhaps even reversed? For centuries, humanity has been captivated by the idea of cheating death and holding on to youth. We've told stories of magical fountains and concocted mythical elixirs, all in the hope of finding a cure for aging. It sounds like pure fantasy, doesn't it? Yet, what was once the realm of legend is now becoming a serious subject of scientific inquiry. Today, in laboratories across the globe, researchers are not just asking if we can slow the aging process, they are discovering tangible ways to do it. This is no longer about simply living longer, but about living better for longer. The goal here isn't necessarily about achieving immortality or living to be 200 years old, though some may dream of that. The more immediate and practical ambition is to increase our health span. Think of it this way: your lifespan is the total number of years you live, but your health span is the number of years you live in good health, free from chronic disease and disability. What if we could extend that period of vitality so that our later years were as active and vibrant as our middle ones? By tackling the root causes of aging itself, scientists believe we can simultaneously combat many of the diseases we associate with getting older, such as heart disease, dementia, arthritis, and cancer. The scientific community now views aging not as an inevitable destiny, but as a biological process that can be understood, manipulated, and potentially even treated. It's a radical shift in perspective. This is the world that today's anti-aging research is striving to create. It's about adding life to our years, not just years to our life. One of the most fascinating areas of aging research sounds like something from a science fiction film: the fight against zombie cells. Officially known as senescent cells, these are cells that have stopped dividing but refuse to die. They just hang around in our tissues like uninvited guests who won't leave the party. As we get older, these zombie cells accumulate throughout our bodies. The problem is, they aren't just idle bystanders. They actively secrete a cocktail of inflammatory chemicals that damage the healthy cells around them, contributing to a state of chronic, low-grade inflammation that fuels many age-related diseases. Think of it like having a few rotten apples in a barrel. Over time, their decay spreads and spoils the rest of the fruit. For years, scientists suspected these cells were bad news, but the challenge was figuring out what to do about them. Could we selectively remove them without harming healthy cells? The answer, it turns out, is a resounding yes. The breakthrough came with the discovery of a new class of drugs called senolytics. These clever compounds are designed to hunt down and destroy senescent cells specifically, leaving their healthy neighbors unharmed. In remarkable studies on mice, the results have been nothing short of astounding. When older mice were treated with senolytics, they showed incredible signs of rejuvenation. Their fur grew back thicker, their kidney function improved, their hearts became stronger, and they could run further on their treadmills. Most importantly, their health span was significantly extended. They didn't just live longer, they lived healthier for longer. This has, of course, sparked immense excitement about the potential for humans. At the very tips of our chromosomes, we have protective caps called telomeres. You can think of them like the plastic tips on the end of a shoelace that prevent it from fraying. Every time one of our cells divides, these telomeres get a tiny bit shorter. Over a lifetime of cell divisions, they gradually wear down. When they become too short, the cell can no longer divide safely and either dies or turns into one of those troublesome zombie cells we just discussed. This progressive shortening of our telomeres is considered one of the fundamental hallmarks of aging. It's like a built-in biological clock, ticking down with each cellular generation. This discovery, which earned a Nobel Prize in 2009, opened up a tantalizing possibility. What if we could stop our telomeres from shortening, or even lengthen them? If we could protect these chromosomal caps, could we extend the lifespan of our cells, and, by extension, ourselves? The key to this lies in an enzyme called telomerase. This remarkable enzyme has the unique ability to rebuild and lengthen telomeres, effectively turning back the cellular clock. In most of our adult cells, telomerase is switched off, which is why our telomeres shorten as we age. But in certain cells, like stem cells, it remains active, allowing them to divide for much longer. In studies with mice engineered to lack telomerase, the effects of aging were dramatically accelerated. They developed gray fur, frail bones, and failing organs much earlier in life. But when the researchers reactivated the telomerase enzyme in these aged mice, something incredible happened. The mice were rejuvenated, their shrunken organs regrew, their cognitive function improved, and their telomeres were restored. It was a powerful demonstration of the direct link between telomere length and biological age. However, this approach comes with a significant note of caution. There is a dark side to telomerase. Cancer cells also use it to achieve their own form of immortality, allowing them to divide uncontrollably. Activating telomerase throughout the body could potentially increase the risk of cancer, a trade-off that researchers are working diligently to navigate. Imagine if your body ran on tiny, rechargeable batteries. In a very real sense, it does. Every single one of our cells relies on a crucial molecule called nicotinamide adenine dinucleotide, or NAD+, to function. This coenzyme is absolutely vital for life. It plays a central role in metabolism, helping to convert the food we eat into energy. It's also essential for repairing damaged DNA and keeping our cellular defense systems in working order. Essentially, without NAD+, our cells would quickly run out of power and our bodies would grind to a halt. The problem is that, as we age, our natural levels of NAD+ decline dramatically. By the time we reach middle age, we may have only half the amount we had in our youth. This decline is thought to be a major driver of the aging process. As NAD+ levels fall, our cellular energy production falters, DNA repair becomes less efficient, and our ability to fend off cellular stress diminishes. This energy crisis at the cellular level contributes to muscle weakness, cognitive decline, metabolic problems, and a general loss of vitality. This discovery has led to a huge surge in interest in finding ways to boost our NAD+ levels as we get older. Since taking NAD+ directly as a supplement isn't very effective, researchers have focused on its precursors. Two of the most studied precursors are nicotinamide riboside, or NR, and nicotinamide mononucleotide, or NMN. When we consume these compounds, our bodies can convert them into fresh NAD+. The hope is that by replenishing our dwindling supply, we can recharge our cellular batteries and counteract some of the effects of aging. The results from animal studies have been incredibly promising. When older mice were given NAD+ precursors, they showed remarkable improvements in health. Of all the breakthroughs in anti-aging science, perhaps none is more mind-bending than cellular reprogramming. This is the idea that we can take an old, specialized cell, say a skin cell from an 80-year-old, and turn back its developmental clock, making it behave like a young, vibrant stem cell again. This concept was once pure science fiction, but it became a reality in 2006 when Japanese scientist Shinya Yamanaka made a Nobel Prize-winning discovery. He identified just 4 specific proteins, now known as Yamanaka factors, that could wipe a cell's memory and reset its identity. This process, called partial reprogramming, is like performing a factory reset on a cell's operating system. It erases the epigenetic marks, the chemical tags that accumulate on our DNA over time, and dictate how our genes are expressed. These marks are a key part of how a cell ages. By removing them, scientists can effectively make a cell young again. In the lab, this has been done successfully for years, but the big question was whether it could be done safely inside a living organism to rejuvenate entire tissues, or even the whole body. The first major proof of this came from a landmark study on mice with progeria, a genetic disease that causes rapid premature aging. Scientists carefully engineered these mice so they could activate the Yamanaka factors for short periods. The results were staggering. The mice that received this reprogramming therapy lived 30% longer than the untreated ones. Their organs, including the spleen and pancreas, showed clear signs of rejuvenation, and their cardiovascular health improved. Crucially, by administering the factors in short bursts, the researchers avoided the risk of the cells losing their identity completely and forming tumors, which is a major concern with this technology. While we are still a very long way from using this technique to reverse aging in humans, these experiments have opened a door to a future we could barely have imagined a decade ago. As we age, our bodies often enter a state of chronic, low-grade inflammation. Scientists have even coined a term for it: inflammaging. This isn't the acute inflammation you get from a cut or an infection, which is a healthy and necessary immune response. Instead, it's a persistent, smoldering fire that silently damages our tissues over decades. This background inflammation is driven by multiple factors, including the accumulation of those zombie cells, a less precise immune system, and lifestyle factors. It is now recognized as a major contributor to nearly every chronic disease of aging, from arthritis and atherosclerosis to diabetes and dementia. A key player in this inflammatory process is our mitochondria. These are the tiny power plants inside our cells, responsible for generating the energy that fuels everything we do. Over time, these mitochondria can become damaged and dysfunctional. As they falter, they not only produce less energy, but also start to leak reactive molecules called free radicals, which cause oxidative stress and damage to the cell. This damage, in turn, can trigger the very inflammatory signals that define inflammaging. It becomes a vicious cycle. Inflammation damages the mitochondria, and damaged mitochondria cause more inflammation. Protecting these cellular powerhouses is therefore a critical strategy for healthy aging. A number of compounds and lifestyle changes have been shown to support mitochondrial health. Coenzyme Q10, for example, is a vital component of the energy production process within mitochondria, and also acts as a powerful antioxidant. Our natural levels decline with age, and supplements are being studied for their potential to protect against age-related decline. Similarly, practices like regular exercise have been proven to stimulate the body to create new, healthy mitochondria, a process known as mitochondrial biogenesis, which helps keep our cellular energy levels high. As we stand on the cusp of a medical revolution, it's clear that the war on aging is being fought on many fronts. We've seen how scientists are clearing out zombie cells, protecting our telomeres, recharging our cellular batteries with NAD+, and even reprogramming cells to a younger state. Each of these strategies targets a different aspect of the complex biological puzzle of aging. But perhaps the most powerful tool to emerge is gene editing, particularly the technology known as CRISPR. This acts like a pair of molecular scissors, allowing scientists to make precise changes to our DNA with unprecedented accuracy. The potential applications for aging are immense. Many aspects of longevity are influenced by our genes. By studying people who live exceptionally long and healthy lives, scientists have identified specific gene variants that seem to offer protection against age-related diseases. With CRISPR, it may one day be possible to edit our genes to mimic these protective effects, essentially giving us the genetic blueprint for a longer health span. This is still highly experimental and raises profound ethical questions, but it represents a fundamental shift from treating the symptoms of aging to correcting its root causes at the most basic level of our biology. So, what does this all mean for us today, on August 10, 2025? It's important to separate the solid science from the hype. Many of these incredible discoveries are still in the early stages of research, primarily in laboratory animals. While NAD+ boosters and certain dietary approaches are available now, treatments like senolytics and cellular reprogramming are still in clinical trials and are likely years away from being a routine part of your doctor's toolkit. The journey from a breakthrough in a mouse to a safe and effective therapy for humans is long and rigorous, and rightly so. We must be patient and allow the science to mature. Yet there is undeniable cause for optimism. The pace of discovery is accelerating like never before. The understanding that aging is a malleable process, not a fixed destiny, has fundamentally changed the game. We are moving from a world where we simply managed old age to one where we can actively intervene in the biology of aging itself. The quest for the fountain of youth may still be a myth, but the pursuit of a longer, healthier life is now firmly in the realm of science.